And can you hear me?
Yes, go ahead.
Yep.
Okay. I am pursuing my PhD in advanced material and with Professor Shah. So currently I am with this group for last 2 years, and this group is mentored by Professor Mistry, Farooq Mistry. So today's seminar, before I embark into today's seminar, first let me welcome Dr. Preston, who has accepted our invitation to be as a guest speaker for today's seminar. Before we go into his biography and the abstract of today's talk, let me talk a little bit about what AME GSC is all about. First thing, AME GSC is having two co-chairs, which is Roshan and Rodney. We have a dedicated publicity and a support team, as well as a correspondence team, which works dedicatedly in this arena such that we can flourish within the campus and outside the campus, and we can reach all around the world. And this group is mentored by Professor Farooq Mistry. Now, AME GSC has his representations in all the arenas within the campus, among all other committees and communities that OU is having currently. So for that, we meet regularly at on every Friday. Right now, we are meeting over Zoom, and you can also be a part of this meeting. Only thing, you have to contact Rodney, who's email Rodney and Roshan, and they will set you up for attending this meeting. If you want to be a part of AME GSC, you can scan this code right now, log in with your OU ID, and you can be a part of AME GSC. Now, what is so good about AME GSC? Now, AME GSC hosts an array of things within and outside campus. Within campus, it's host like seminar series every week. With it could be Wednesday or Thursday. Apart from that, we host spring picnics. Then we go ahead and bring in guest speakers from NASA's, Toyota's, and various organizations where you can find your employment with. And we also host like international food festival in fall. Apart from that, they are on Facebook as well as YouTube. Our Facebook page is this. You can click onto this Facebook page and you can follow us. And the upcoming seminar is about optical and thermal enhancement of concentrating solar collectors, which will be hosted on 19th November 2020 by Mohammad A. Hassan from Cairo University, Egypt. Now, coming back to today's speaker. Now, first, let me welcome Dr. Preston. Now, thank you, Dr. Preston, for accepting our invitation to be our guest speaker in this seminar. Now, getting back to his biography, Dr. Preston is a research associate at Samuel Robert Noble Microscopy Laboratory at University of Oklahoma. He has obtained his undergraduate degree in engineering physics and his master's also in engineering physics and PhD also in physics from University of Oklahoma. He is currently specializing in the area of electron microscope with particular emphasis in high resolution scanning electron microscope of nanostructure and nanotechnology related material. His interests include characterization and investigation into noble physics, originating from well-ordered array of nanostructures, including dots, holes, and rings in metal, semiconductors, and magnetic material and superconductors. Now, I like to add one thing because I have taken one of his course, which is Scanning Electron Microscopy, which is one of the fabulous course. Now, with this, I'll tell you that we, as an engineer, try to build things on engineering skills, but we tend to forget that the behavior at nano level dictates what we see at engineering skills. Now, in order to understand a any of the component in a holistic manner, it's always better to look at the nanoscale to find an answer. So Dr. Preston has helped me in understanding that, both through his courses that he offers. I have taken his scanning electron microscopy courses, which he offers, and that was a fabulous course. I have learned a lot. So I urge all my grad students who are working with material and who are doing experiments and all, I urge them to take his course. And that helped me to understand my material, why it performs like this, why the result I'm getting, what is the reason behind it. So that's a fabulous things that I have learned from him. And today's abstract, if I'm going to talk about that, Dr. Preston will focus on the application and technique of electron microscope and the resources that OU is having. This includes both scanning electron microscope, which we call SCM, transmission electron microscope, which we call TEM, and associated analytical capabilities. Users of these facilities also can work with the dedicated staff for sample prep and analysis. And also he provides hand-on training through one-on-one session or through talking about electron microscope courses offered through the facility. So I will welcome Dr. Preston. Dr. Preston, you are welcome. The stage is all yours.
Super, thanks. Let's see if I can share my screen here.
But you have to stop sharing.
Okay. All right, so hopefully everyone can see my screen now.
Yes.
Super. Okay. Well, I appreciate the invitation to talk today. And as Anirban mentioned, I'm going to try and give you an idea of what sort of characterization techniques we have at the microscopy lab here on campus at OU. So if you're a researcher, grad student, postdoc, whatever, and you need microscopy, you can come to the Samuel Roberts Noble Microscopy Laboratory to get your work done or facilitate your research. There's 4 of us, so it's kind of a small group at the moment. There's our director, Dr. Andy Madden. He's actually a geologist and just took on the role of director of the lab here about a year ago. And he's doing a great job. So we're very excited because we're trying to kind of broaden our horizons a bit and look for, as always, new equipment, right? So we've got ideas for new SEMs, TEMs, light microscopes, you name it, right? This is Dr. Ting Ting Gu. She's our advanced light microscopist. Julian Savish here. We just hired him about a month ago. He's has a material science background and is a sort of a TEM specialist. And of course, here I am at the lower left, working with electron microscopes as well. So if you haven't been to our facility, we're kind of tucked away in between George Lynn Cross Hall, which is the plant biology and microbiology building, and Richards. So you can kind of see the sign here. We're tucked back around here. And actually we're kind of spread out because we're on the 2nd floor of what is essentially a mobile home type structure. It's not a very good place to house microscopes, but that was what was available in the late 80s when this was constructed. And so we've kind of branched out and we've got microscopes both in George Lynn Cross Hall in the basement, as well as light microscopes, Ting Ting's area in Richards. So you can see we've got, at the moment, 3 TEMs, 3 SEMs, and some confocal microscopes. And we're looking, again, as I mentioned, to branch out, hopefully in the near future. And so what I'd like to do is kind of give you an idea or an overview. Primarily, I'm going to focus today just on the electron microscopes. I'm going to neglect the optical microscopy side for today. Perhaps Ting Ting can come and give a talk to you guys at some point if there's interest, which is not to in any way take away from the wonderful stuff that she does. In fact, Anirban was at our lab, I don't know, a couple weeks ago, I think, to do some electron microscopy, and it turned out doing some light microscopy was actually better for the results he was looking for with his 3D printed structures. So that's neat. Regarding microscopy just in general, I know everyone knows what a microscope is. And so SEMs and TEMs, they're a little more technically complex, but they're designed to do the same thing that an optical microscope is. So produce a magnified image of the specimen, and more importantly, resolve finer features that you can't see by eye. The ultimate goal, obviously, being to understand your material better, learn about it, manipulate or control it, or whatever the research goal you have. And so this is a very early Van Leeuwenhoek microscope shown here. Pretty simple in design, but it was one of the original microscopes, light microscopes. So you can see a little glass lens here, sample holder, which is nothing more than a pin to attach your specimen on. Focus knob, which just adjusts the height between the sample and the lens. And you'll notice there's no light source, so you would use ambient light, either sunlight or candlelight or something like that. So light microscopy is great, and as I mentioned, in some cases it's the go-to technique for what you need. But one of the issues, and sort of a motivation, if you will, for moving to an electron microscope is the resolution limit. And so I'm not going to go into all this today, but light is a wave, and so it will scatter off matter, and that's called diffraction. And that will actually limit the resolution that you can obtain. Whoops. There's a very famous equation in the microscopy field. It's called Abbe's equation, and it describes the diffraction limited resolution. That's sort of the ultimate physics limit for resolution, the best you can hope to obtain. And I notice my equation is not showing up fully on this screen. I tried it on my phone and on my Windows laptop. I'm on an iPad now, so I wonder if there's some compatibility issues. But anyway, it's a famous expression, and it's very simple. Basically, it says your resolution here, which is D. So in other words, if you want to resolve finer things, you'd want this to be as small as possible. It depends on a constant here, but then what's not shown is a lambda, the Greek letter lambda, which is the wavelength. And so for optical microscopy, we're in the hundreds of nanometers wavelength here. And then something called the numerical aperture, which is n sine alpha. And you can see that alpha again is missing. N is the index of refraction, which typically is 1 if you're imaging in air. But if you've ever used an oil immersion lens system, for example, oil has an index of refraction higher than 1. And so if you utilize that, you can gain or get a smaller number here, gain in resolution. And then sine alpha is the half cone angle of a lens. But the end result is that if you plug in all the numbers for light microscopy, you're limited to somewhere around 200 nanometers, which is really incredible. But there's more room at the bottom, as Richard Feynman said, and so you could imagine going down ultimately to maybe atomic resolution or beyond. And so that's something that EM can give you. Before I move totally away from LM, I just thought I'd give a very brief history, and it's incredibly brief. So basically there's stuff before 1590. There's many ancient civilizations that had some sort of optical principles that they were aware of and actually utilized. But it's considered to really get its start historically late 1500s, early 1600s with these guys. Hans and Zacharias Janssen, they were spectacle makers or glass makers, makes sense. Robert Hooke, who had his hand in all sorts of stuff, as they all did in those early days, and Van Leeuwenhoek, who I mentioned before. Robert Hooke wrote a book, Micrographia, where this illustration, very highly detailed illustration of a flea came from. And so this was a book that described his findings with his microscope in very high detail. And in fact, we have a copy of Micrographia in the History of Science collection at the library. So if you ever get a chance to see that, check it out. It's amazing. I got to see this actual illustration in the book itself, for example. So, in terms of where it went from there, so basically the early work was involved with trying to make these glass lenses and making superior glass lenses. In fact, Van Leeuwenhoek was known for the quality of his lenses. Again, this is another image of one of his microscopes. Then sort of in the 1870s to early 1900s, they started to create science or math. They started to get a better understanding around the optical principles. So for example, Abbe's equation, which tells you that the resolution you can hope to obtain with an optical microscope is actually dependent, for example, on the wavelength of the light that you're using. So you can see here, that's what Carl Zeiss was trying to utilize, that principle when he created a UV microscope. He tried to create a microscope with incident radiation that was a smaller wavelength, right? And you can only take that so far, I might add. You could say, okay, well, let's go to X-rays and things like that. And in principle, in terms of the wavelength, that's totally valid. And there obviously are X-ray systems out there, MRIs and such like that, imaging systems. But it's hard to focus X-rays. They're known obviously for their penetrating power, and so creating a X-ray microscope with a resolution that's appropriate to what its wavelength would dictate is a challenge in and of itself. And so hence the motivation, for example, for moving to maybe electron microscopes. So electrons, if you're considering them as a particle or a wave. So in early 1900s, quantum mechanics came around and you could associate both particle and wave-like properties to electrons. And so it turns out that right around that time, they said, oh, maybe we could use this instead. In fact, the wavelength is totally dependent on its energy. And you can give an electron energy, for example, by accelerating it through an electric field, so applying a voltage. And so that means you have a tunable, in some sense, wavelength of an electron that you could use. And that's essentially what they did. So they just used the de Broglie relationship and came up with Abbe's equation as it pertains to electron microscope. Again, I can see my equation didn't quite show up. Hopefully it's correct on the slides. There should be an equal sign here and an alpha, that half cone angle here. But you'll notice the V is the voltage. And on any electron microscope, you'll see one of the primary controls or knobs on the microscope will be the accelerating voltage. And that's directly related to setting the electron wavelength. And so you can see as you go to higher accelerating voltages, here's 100 kV, that's a typical TEM type accelerating voltage, 200 kV, 300 kV, up to even 1,000,000 volt. That used to be a thing in 80s, 90s. They've sort of moved away from that with the advent of aberration corrected microscopes. But the idea is that you get better resolution, as you can actually see here. So that's summed up nicely in this sort of chart here, which shows the length scales involved and what you can do with your eye. And of course, with your eye, it's going to depend on how close the feature is, and people's eyes are different, but it's typically limited to somewhere around 1/10 of a millimeter or 100 microns, which conveniently is about the size or thickness of a human hair here. LM, light microscopy, as I mentioned, can get us down to about 200 nanometers or so before the diffraction limit sets in. But that's good enough to see things like cells, for example, bacteria, things like that. But if you wanted to see structure inside the cell or features on the bacteria at a smaller length scale, the light microscopy is going to come to a hard limit. And that's where EM comes in, both in the form of SEM or TEM. SEM is typically, it can go down to about 1 nanometer. There are specialized cases where an SEM is shown full atomic resolution, but that's a very specialized case. Typically, 1 nanometer nowadays is kind of where it caps off. And then the TEM, especially with what are called aberration corrected TEMs, which is a fancy term for correcting a lens defect, an aberration. They can now get down to incredible resolutions measured on the order of tens to hundreds of picometers. So fully into the length scale of an atom, so atomic resolution. And so I like to show this image a lot because one, it was taken with our TEM here at OU. And two, it's not necessarily a pretty image. You can see it's kind of noisy. What it is a specimen of silicon along a certain crystalline plane. It happens to be the 110 plane. And along that particular plane, the atoms line up in what are called dumbbells, or actually two atoms side by side. And so you can just, with our microscope, resolve that. And you know, is this just a cylindrical structure, or is this actually atoms? In this case, it is actually atoms, and we're right at the resolution limit to where you can just say, hey, there's two structures here. You can do FFT filtering to remove some of the noise, and it becomes a little more clear. But essentially, those atoms are spaced about 1.3 angstroms apart. And so you're fully on the atomic scale at that point. And I, again, will mention that this is, you know, really good, but the state-of-the-art microscopes, which we're hoping to figure out a way to get, can push you down into the tens to hundreds of picometers, so even further. So in terms of a very brief history of EM, basically starts with Abbe's equation that showed that the resolution depends on the wavelength, okay? And then the quantum mechanical revolution came. They discovered the electron. De Broglie associated, hey, this can have a wavelength, so maybe we can use it as radiation for an imaging system. The question then becomes, well, then you have to have some sort of lens to magnify your image. And that's where it kind of falls apart from LM, where you're using refraction or using curved glass surfaces or plastic surfaces to focus light. Here, they take advantage of the fact that electrons are charged particles, so you can use magnetic fields or electric fields. And basically, if you've heard of electromagnetic lenses, that's what SEM or TEM uses. And this was developed by Bush and Ruska in the late 1920s. In fact, Ruska wins the Nobel Prize, or shares it, in 1986, so quite a bit later. And by all accounts, he was pretty, and probably rightfully so, he's kind of a little frustrated that it took that long for him to win the Nobel Prize, and he shared it with the people that invented scanning probe microscopy techniques like STM. You can see an early replica of his TEM here. And while it does look fairly primitive, this basic design will hold. And so you'll see microscopes today that have a central column, and that's essentially where the electrons are generated up at the top and they come down the column to interact with your sample. So historically, TEM, transmission electron microscopy, came first. SEM as a concept was right around that time, but you don't see a commercial SEM until much later, 1965. And so you can see some early SEMs here. And so in some sense, SEM got a much later start in life. And as I mentioned, the concept was there, but the problem was they didn't have a good enough detector to detect these scattered electrons coming off your sample. And there's a very famous Everhart and Thornley SEM detector that's to this day on almost every SEM that circumvented that problem. And really, the Everhart-Thornley detector is fairly simple in and of itself, and the main part of it is just a photomultiplier tube. So anyway, taking us down to 2000s, 20 years ago, you're now to a point where people are just improving lenses and sources, electron sources, everything about the systems. And we're getting down to, again, in some cases, atomic resolution on an SEM and subatomic resolution in the tens to hundreds of picometers on a TEM. So pretty incredible. So our lab has TEMs and SEMs. TEMs historically came first. And so what I'd like to do is just take you through a bit of a whirlwind tour of what a TEM does and all the applications that you can do with it. So maybe you could figure out or an idea for your research or how it would benefit your particular materials that you're looking at. So TEM, operative word is transmission. So that implies that the electrons have to transmit through the sample. And that has implications for sample prep, obviously. So, for example, it could be very simple if you are a researcher that is looking at nanoparticles. Well, they're already really thin. You can just put them onto a TEM sample grid and directly look at them. But if you have a bulk sample, if you're a geologist, you're looking at a rock, or you're, we've got electrical engineers that grow semiconductor structures that are fairly thick. How are you going to look at that? Or if you're a biologist, for example, and you want to look at a cell, how are you going to do that? Because the cell is fairly small, but it's microns to tens of microns, but you need something much, much less than that. And so even half a micron here would be really too thick. A gold standard for a biological specimen would be like 70 nanometers. For materials specimens, that could be smaller still, down to atomic or a couple atomic layers. So sample prep is an issue, and I'm not really talking about it today, but I'll just give you the quick idea. If you've got a polymer or a biological material like cells, you'll embed it in a polymer, and then you'll take very, very thin slices with what's called an ultramicrotome, which consists of a diamond or a glass knife, and you're just shaving off very, very thin layers of material. If you're a geologist or a semiconductor researcher with much harder material, you may be mechanically polishing it down and then following that up with some sort of ion etching to etch through your material. The other thing that I should note that it's not explicitly expressed on the slide is this microscope, as you can see here. This is the column. The sample has to be input right around here. That's the sample entry. This is all done under a vacuum. And so that has consequences as well. And so you've got to suck all the air out using vacuum pumps. The idea behind that is that the electrons will scatter off air molecules, and you want it to scatter off your sample. So you got to get rid of all the air molecules. And that's not an issue so much for material researchers, but again, people like biologists, for example, where you're looking at cells that are much more natural to be in a wet or aqueous type environment, and now you try and put it into a vacuum. You've got, you know, you kind of, it shrinks up and distorts and all that sort of stuff. So typically you have to worry about that as well. And the solution there is typically some sort of chemical fixation, glutaraldehyde or formaldehyde or something like that to preserve the structure. Anyway, if we take a look at a little video here. So here, let me pause it maybe. So just up top was the source of electrons, and that's either a heated filament, like a tungsten filament that heats up and produces electrons, or it could be a field emission source where you use an electric field to drag electrons off. Then the electrons go down here, and what you're seeing here is a cross-section of a lens, an electromagnetic lens, and so these little black dots are meant to illustrate copper wire that is wrapped around in a loop, okay? You apply a current through that wire, you produce a magnetic field, and the magnetic field is what is focusing the electrons. And here you're basically interacting with the sample. So TEM, I always like to give the analogy that it's very similar to when you have a flashlight and you take your flashlight and shine it on your hand, for example, and then you can see the shadow image on the wall. That's essentially what you're doing with a TEM, and then you're using lenses to magnify that shadow image. In fact, it's directly analogous to a light microscope. So if you understand how a transmitted light microscope works, you understand the same principles are working with a TEM, except you're switching out the light for electrons. So, although in some cases you could use a tungsten filament for both. If you heat a tungsten filament, for example, you can get light out. If you heat it a little hotter, you can get electrons out. Don't have to use that as a source, obviously, but that's something you can do. Then, of course, you have to have lenses, and you use glass lenses for LM, electromagnetic lenses, as I said, for TEM. The parallel incident beam shines onto the sample, and you get this shadow image that you then magnify with lenses, and then you have to view it either with your eye or a camera. So, okay. And so that's what's called bright field imaging, shown or expressed right here. But that's just a small bit of what an TEM can do. There's a lot of imaging modalities. So you can see here, bright and dark field imaging, high resolution, TEM, scanning, or STEM as it's called, electron diffraction, analytical techniques like EDS and EELS, and Cryo Tm. So this is what I mean by like a smorgasbord. And what I want to do is just give you sort of like a slide on each. And I know this is a lot of information, but hopefully, you know, at least my idea is to let you realize what sort of capabilities are available, so that can get you started thinking about what this could do for example, your research. So, bright field imaging, that's your kind of conventional TEM image, and you can do it with nanoparticles as shown here, or biological materials. Here's the cell shown here. Basically, you are looking at the, or your signal is
Has he disappeared?
I think because of some internet issues.
Yeah, okay, so let's just wait.
It's weird that it didn't kick his user off of the Zoom meeting. I wonder if he knows that we can't hear him anymore.
Means he's speaking, he also don't know that we are not able to hear him, right? I mean, we are also speaking, right?
hello.
Yes.
Hi.
Hi.
Hi. Sorry, guys. I don't know if that's my hotel Wi-Fi. I don't exactly know when I left off. I just started hearing a buzzing, and so I asked if anyone was there, and I didn't get a response. So, but you guys can see me now, right?
Yeah, we can see
Okay.
you and hear you.
Okay. Well, hopefully that won't happen again, and I'll just continue. So sharing my screen here. Okay. Okay, so I don't exactly know where I left off, or where the transmission got cut off.
Bright field.
Okay,
Yeah.
good. So there's bright field. So for example, these are gold nanoparticles that were taken on campus by a biomedical researcher. So they were interested in sizes and shapes. And if they're easily they're dispersed like this is actually very nice. You can do something like ImageJ or other software packages to do the heavy work for you and get out the sizes and shapes. In fact, I could just give a little plug. We have a little Focus Friday seminar in the microscopy lab through Zoom, a Zoom meeting. Anyone's welcome to join. And this Friday, for example, Dr. Gu is going to be talking about analysis using ImageJ. So you might, if you're interested, you could check that out. There's also what's called dark field imaging, which the idea behind dark field imaging is to look at the scattered electrons. So don't look at the unscattered, but look at the scattered component. So this circle here is an aperture in your microscope. And so if you center it around the unscattered part of the beam, that's what's typically called bright field. If you were to move your aperture and look at the scattered components, that would be called dark field. And that's useful for material scientists who are looking at crystalline structures or maybe high atomic number type structures. So for example, here's a bright field image of carbon nanotubes. Here's the dark field image, and the little catalyst particles are glowing or shining bright. So You can take it even further, and again, down to the full atomic level, and this is usually considered a different modality or different imaging mode, because you're trying to collect not only the unscattered component of the beam, but as many scattered components as possible. They will all combine. This is called phase contrast, and they'll all combine, and it involves constructive and deconstructive interference, and what you get is a series of bright and dark spots on your screen that you can, or on your image, that you can interpret as atoms. So you can get positions of atoms. You can look for defects, for example, in a crystal. You can get lattice parameters, the spacing of atoms, things like that. And I should mention that this is quite, it's a little more complicated, because depending on your focus, just a little subtle change in the focus can make the bright spots go dark, or vice versa. And so you really don't know what is the actual atomic position sometimes. And sometimes that's okay. That's good enough to see defects. But if you do really want the actual structure that you're looking at, you have to combine this with image simulation. So you have to put in your imaging parameters into software, and it will simulate knowing the exact amount of defocus that your microscope is under, and the material properties, and then you can confirm that this is indeed the right proper structure. So then there's STEM, Scanning Transmission Electron Microscopy, and this is very analogous to SEM, actually, because scanning electron microscopy. And so in this case, you're not trying to come along with a parallel beam on your sample. What you're actually doing is you're taking your beam and focusing it down to a small probe or a point. And so you're probing one particular spot, and then you move over and probe another spot, and then another and another, and that's the scanning part of it. And there's there's a number of advantages and disadvantages between using a TEM in conventional TEM mode versus STEM mode. And so there's some cases where it's better to use STEM. STEM is better, for example, for thicker samples and other maybe damage. It reduces damage because you're not on any one spot of the sample for very long, whereas with bright field TEM, the beam is continuously shining. And while we consider it non-damaging, that's not actual reality. That electron has a bit of energy and can actually damage your sample, especially prominent with softer materials like polymers or biological materials. So you can also do what's called diffraction. So diffracting off of crystalline materials, and you can get all of these gorgeous diffraction patterns that are quite complex and rich with a lot of information. So it can tell you, for example, orientations of your crystal, or again, lattice parameters. This involves Bragg diffraction. It can give you the distance between atomic planes. And so analyzing these diffraction patterns can be a bit complex, as you might imagine, but it's very simple to get a diffraction pattern. You can get one in minutes and very quickly, for example, in a matter of seconds, tell if it's polycrystalline, or if it's a single crystal, or if it's amorphous, just by looking at the diffraction patterns. So that's something that's very useful for a lot of people as well. Another thing you can do, since you're irradiating the sample with electrons, and typically you're looking at the electrons that scatter or don't scatter out of your sample, but some electrons also excite X-rays. So if you look at this little movie here, your primary beam comes in on your conventional atomic model, and you can knock out electrons in their orbitals, and higher electrons in higher energy states can drop down. As they do so, they can emit X-rays. So if you were to have an X-ray detector and you detected this X-ray, for example, coming off, measured its energy, then you could determine what atom you interacted with. So basically it's an elemental identification technique. And so you get a spectra that looks like this. And so you can see here, here's a chrome peak, copper, tantalum peak. And so it allows you to do a little bit of understanding the chemistry, if you will, of what's in your material, and you can map for it too. So here's a TEM image and resulting TEM maps. This was kind of a neat little project that someone was that worked on number of years ago, it's no longer here, with a plant, and the little dark spots are silicon oxide, silicon nodules, and silicon and oxygen maps light up for the nodules. So those are little silicon oxide particles. Another analytical technique that you can do with a TEM, and we actually, I want to be clear, we had this technique up until about half a year ago, and we had to get rid of it for an upgrade for a digital camera to make room for a digital camera. But we're hoping to get this technique back in the near future. As I mentioned, we're exploring other options for other microscopes. But there's something called EELS, which stands for Electron Energy Loss Spectroscopy. And I know this is a complicated looking diagram, but essentially what it's doing is with TEM, your electrons come in, and if it scatters off your material, if you could somehow measure how much energy that electron lost, then you could work back how it must have scattered. For example, it must have scattered off a carbon atom because it lost this amount of energy, or it must have scattered off a copper atom or whatever. So you can get a spectrum in some ways very similar to what I showed before with the EDS, where you're measuring the energy of X-rays. Here you're measuring how much energy the electrons have lost if they've scattered through the material. And while it can give you similar information to EDS, it can give you much more information as well. And so there's very complicated LNS, Excels, extended energy loss, and near the energy shell. And so it's kind of complicated, but it can give you more information on things like oxidation states, Fermi densities, things like that. And so it's quite a powerful technique as well in its own right. And you can do that on a TEM that's equipped with an EELS spectrometer. So an EELS spectrometer is just basically a way to take the electrons and disperse them according to their energy so you can get the spectrum. So very useful technique. Another very useful technique that we're starting to explore is what is called Cryo Tm. So it's new to us, but it's not new to the world. In 2017, it won the Nobel Prize. You may have heard about this in chemistry. And the technique involves, remember, you are in a vacuum environment in the TEM. And so you can't look at water, for example, which a lot of things like proteins or viruses or whatever might be living in, right? But if you could freeze the water, then you could look at that. So the idea is you have to freeze, and that requires its own state-of-the-art equipment because you don't just put it into the freezer and take it out 4 hours later and look at that ice. You have to freeze it very, very quickly because you want amorphous ice. And if you don't freeze it very quickly, it'll crystallize. But the idea behind it then is if you can take something like a protein structure in water and freeze it, and then put that in the TEM and keep it cold. So you have to have a stage in the TEM that keeps it sort of at liquid nitrogen temperatures. Then you could look at, for example, proteins. And you see here, there's a number of proteins. They're all the same protein, but they're in different random orientations. But if you take enough of these images and use computer processing power, you can start to classify the different orientations they're seeing, and you can actually work out a 3D structure. And so this kind of revolutionized the field. They were relying on other kinds of techniques for getting protein structures, and now Cryo Tm is sort of taken over and is getting resolutions that are pretty incredible for proteins. So, and of course you can use this on other structures as well. This is some of our work, again, done by Dr. Stefan Wilhelm, who's a biomedical researcher here on campus. And it's kind of hard to see with this, but these little tiny circles here are liposomes, basically phospholipids that it can encapsulate a water molecule or water droplet, many molecules. And they can be used for drug delivery and all sorts of different applications like that. So it's kind of neat. But to see them clearly it involves the use of Cryo Tm. So that's kind of what the TEMs can give you. And I thought I'd just give you sort of a virtual tour real quick of the TEMs that we have. So this is our Zeiss 10 TEM. I'm looking at the clock. Notice I'm running over. So I apologize. I'll probably go fast. If anyone has any questions, please I'd be happy to answer now or later. But this is our Zystand. This is, if you took the TEM class, for example, you'd start on this machine. It's 100 kV, perfectly applicable for biological research and very quick and easy. So you can put in a sample and in a matter of minutes, get out an image. Doesn't have any analytical capabilities. So if we move on to our next scope, this is 200 kV. Remember, that kind of tells you that's an upgrade in the resolution. That's kind of code word for a better resolution. And we also have analytical capabilities. This thing hanging off the side is the EDS detector for elemental analysis. And we can also do STEM on this machine, scanning transmission. And then our best, if you will, microscope. This is a field emission microscope and a 200 kV instrument. It's our JOL 2010F. And this is the one that can get down to atomic resolution fairly easily. It has, can't see it, it's off to the side, but it has X-ray EDS as well, has STEM capabilities. And also, I haven't really talked about detectors today or camera systems, but it has a direct electron camera, which is a state-of-the-art imaging system that can actually provide its own resolution bump. Basically, in the past, you had to use film, just like camera in the old school days, and then they moved to CCDs and CMOS. But in CCD and CMOS cameras, you still have to convert the electrons to light. This detects the electrons directly. It's actually CMOS based, but it's detecting electrons directly, which provides a great benefit for the imaging. So, all right, so that's sort of what you can do on a TEM. What about SEM? So SEM, the operative word would be the S, scanning. And so you are taking your microscope, or your electron beam, and rather than trying to irradiate your sample with a parallel beam, you're actually focusing it down to a probe, kind of like the tip of a pencil. Okay, so if I play the little movie here, showing the insides of the an SEM, I'm going to try and pause it maybe right here. So if you can see the little yellow trace here, this is your electron beam coming down at the tip of a pencil onto a sample that would sit here. So the ultimate probe diameter is going to vary depending on your settings, but the idea is the smaller the probe, the better your imaging resolution. So you can get down to nanometers in probe size. And then as it hits your sample, it's going to scatter, and scattered electrons are going to come out. And then you have various detectors. Here's the Eberhardt-Thornley detector that will detect what is called secondary electrons, which are electrons that are being emitted from the sample. So they're not the primary beams. The primary beam comes in and kicks off sample electrons, so they call them secondaries. Or this detector here is a backscatter detector, and that is a primary beam detector. So in other words, the primary beam comes in and it can scatter or bounce back. And so you can detect those signals. This little finger coming in here is for the X-rays. So for doing EDS, just like you can do in a TEM. So again, this is all in a vacuum like a TEM, so you have to worry about sample prep. In addition, your sample has to be conductive because you're radiating it with these electrons, which are negatively charged particles. And if your sample is insulating, then you're going to build up a negative charge on your sample, and you'll actually reach a point where you've got a negatively charged sample and a negatively charged beam coming in, so you'll actually repel the electrons. And so the solution often or typically is just to put a very, very thin, and by thin I mean maybe a few nanometers of a metal on top of whatever your insulating structure is. And that usually is good enough. Maybe it doesn't eliminate the charging, which is the #1 artifact you see in SEM, but mitigates it to the point where you can take reasonable images. So this is another schematic that hopefully shows that maybe in a little better detail. It's a little simpler, but shows this blue cone here. That's your electron beam coming down as a probe. And then if you took a snapshot in time, it scatters on one spot, scatters electrons to a detector, and then it moves over. And that's the scanning part. And so you scan typically a square or rectangular area. And you can develop an image based on the scattered electrons, depending on which detector you're using. So here I've just got a generic detector, but again, you can use secondary electron detectors, backscatter detectors, X-ray detectors, etc. And so I'll quickly go through that. So much like the TEM, you've got different imaging capabilities here. Secondary electron image is probably the what you see in the literature when you're perusing a paper and you see an SEM image. Probably greater than 90% of the time, what you're looking at is a secondary electron image, because the number of secondaries, remember that's sample electrons that are being scattered out. That's a large function of the topography of your sample, okay? So it's good for basically getting an image that is very much like what you'd see with a reflected light microscope, even though the image construction is very different than a reflected light microscope. The image that you get can be interpreted in the same way. So it's often very intuitively interpretable, much more so than a TEM, which is looking through the material, and you've got to wrap your mind around looking through a cross section. This is much more intuitive. For example, this is a flea, right? And this is a flea's head, and it looks exactly like what you'd expect a flea's head might look like under an optical microscope or with your eye, only we're magnifying it a little larger. This happens to be some sort of bacterial cells. Okay, so secondary electrons are good for topography. If you use the backscatter detector, that's good for material contrast. So rather than giving you topography or structure, what it will do is backscattered, you'll get more backscattered electrons if you interact with material that's denser. So in this case, you've got carbon nanotubes, which are, you can see the little ropes here, which are made out of carbon, which are not very dense. You know, carbon is a low atomic number, but then you see these bright spots showing up throughout. So that tells you that those bright spots are something at a higher atomic number, which are the catalyst particles, the metal catalyst particles used to grow the carbon nanotubes. This is another example with, I looked at a meteorite with the backscattered image. And for the most part, the surrounding matrix is aluminosilicate type structures, but there's also metallic iron, and you can see that popping out nicely. Iron's more dense than the surrounding material. So backscattered imaging is used for compositional analysis, or even if you're a material scientist that's involved with crystalline structures, you can get crystalline contrast too, and specifically defects in a crystal. So you can see what are called twinning structures here in a semiconductor material. So also useful for material science researchers who are looking at crystals. If your sample is thin enough, it doesn't have to be, but if it's thin enough and you could put it into a TEM, for example, you can also do transmitted electron imaging by sticking a detector right beneath your sample. So this is essentially STEM, but you could do it on an SEM or a TEM. The difference being that SEM is usually utilizing much lower accelerating voltages, which has consequences in terms of resolution versus contrast. And so there's, again, advantages and disadvantages for doing STEM in an SEM or doing STEM in a TEM, for example. So for one case, you might want to use SEM. For another case, you might use the TEM. You can also do EDS here, so I can go through this quickly. But again, you can get a spectrum that lets you know what elements are in your sample. And so, for example, this is a spectrum taken off this meteorite image that I showed, and it's showing all the elements present within this whole field of view. So, for example, I don't know what this is. For example, I know it's one of these elements, but I don't know that it's necessarily the iron. But then if I do an EDS map, you can see iron being denoted by the kind of bluish color. Iron lights up there. So that's the iron, whereas the silicon, magnesium, oxygen light up the surrounding matrix, so the aluminosilicates. So that's useful for chemical analysis and for actually site-specifically locating where different elements or different materials are. You can also do diffraction in an SEM, electron backscatter diffraction. I gave a little Focus Friday seminar on this just a couple of weeks ago. We have this capability on our Zeiss Neon. And again, it's a way of, if you are a person who is researching crystals, it's a way of getting a diffraction pattern that you can fit to a particular orientation. So for example, this is a hexagonal gold nanoparticle, and it's all lighting up as blue, which means it's the 111 orientation direction facing outward, normal to the plane of the image. And so you can, for example, I've used this with geological applications, electrical engineers, again, semiconductors, things like that. Metallurgy, if you've got grains of different crystalline orientation showing up, this is a technique that can get you that kind of information out. So I'm going to go through this pretty quickly since I'm running out of time here, but just sort of applications. We do a ugly bug. This is an educational outreach sort of project we do where we image bugs that kids send us from around the state of Oklahoma. And we usually get about 100 people, or schools rather, per year participate. And so it's kind of just a neat thing to get kids interested in science, but we do actual research on bugs as well. This is kind of just a fun thing, but this is actual research looking at the compound eye structures of fruit flies and fruit fly mutants, and seeing what genes are involved in taking out the eye structures, for example. Here's a nice picture of a fungus. So this was done a number of years ago by a person here at OU. This is Aspergillus niger. It was the cover of a journal. And I show this just because I can highlight the idea that most SEM images you see, what you're looking at is just a signal intensity, the number of scattered electrons, right? And so typically they show that as a grayscale. So white being a lot of scattered electrons, dark being, or black being no scattered electrons, like down in, say, here, for example. And then grayscales in between to represent that. But you can false color this. And so that's what we've done here. And so when you see that on journals or online, there you can, they're using Photoshop or something like that to false color the image to highlight important details. So it's going to be very useful. But typically you're looking at this in a grayscale image like this. So we do a lot of work. We're in the our labs are associated, we're not associated with microbiology, but our labs share space with microbiology labs. And so we looked at biofilms, all sorts of stuff. This is synthetic bone materials, geology, hematite. Here's weathering of quartz grains. And so if you see gouges or very sharp edges, that's typically inferred to be due to glacial transport. So ice damage, obviously, rather than more typical rounded structures you might see from, water erosion over time. So lots of things like that. Since this is AME, a lot of the AME stuff, I didn't show many examples today, but we're typically, or what I've done for a lot of AME people is to look at composite materials, for example. So some sort of typically carbon nanotube or fibers or graphene or something in a plastic or a polymer. And people are interested in high strength materials or electrical conductivity or whatever, and they want to see how dispersed their filler particles are within the matrix. So do a lot of that. This is a nice example. Again, this is on a quartz grain, but SEM images are two-dimensional. So you can see a scale bar here, for example, this 200 microns, and that's the scale that's appropriate in the X and Y direction. So you can do particle sizing very easily, but it's a two-dimensional image, so you can't tell depth. So for example, on this image, looks like there may be a trench here, a little hole or trench in the grain. And you can infer that from the image, and you'd be right, but what you can't do is you can't measure the height of that. But if you take two SEM images at different tilt angles, so you tilt the sample back and forth and take a couple of images, then you can combine that and get back that missing third dimension. So you can get back a height, for example, and so see how deep that particular trench is in a quartz grain. And this is a nice example where you want to measure heights, and typically you might want to do that with an AFM, an atomic force microscope. But in this case, being a quartz grain, it's a little too rough and irregular to get the height in that manner. AFM really uses, your samples have to be quite a bit flatter. And so this was an ideal case to be able to use the SEM to get back a height profile. So more carbon nanotubes. Again, I've looked at a lot of this with AME people, typically with carbon nanotubes in a material, in a composite material like a polymer. So other stuff on campus is really neat, the kind of mushroom-shaped carbon nanotube forests. You can do heating stages or cooling stages on SEMs. And again, I apologize, I'm just going through this really quickly, looking at the time. And so again, if anyone has any questions, you can hit me up later or at any point. Another neat thing you can do with an SEM is rather than using that SEM as a microscope, as it was designed, you can actually use it as a writing tool. So you can take a material that is very sensitive to electron beam radiation, and it's typically a polymer. And then you can write a pattern with the SEM beam. And so, for example, this is a map of the United States, right? And so you can do cool things like that, or you can do this is an actual research application where they're trying to create essentially a circuit for doing some physics experiments here. And so that's a neat application called electron beam lithography. Our Zeiss Neon has what's called a focused ion beam as well. So you have an SEM as well as a focused ion beam that's available. And what that means is ions are much more massive than electrons. And so when they hit the material, they'll actually damage it. And that can be a good thing because you can then use the focused ion beam again to create patterns like OU or this buffalo here, or to actually cut into material and look at the interior of it. And so combining the fib to do the damage or the etching and then do the imaging with the SEM column, you can have a whole lot of cool applications. So for example, here's a pollen grain, and they wanted, we have a researcher who wanted to look at the interior of the pollen grain, what's called the Axine structure, which is useful for taxonomic purposes, identifying the species of pollen and whatever. And so we used a focused ion beam to just cut it in half. So you can see it's cut in half, and then we can tilt it and look on the side, and you can see the Axine structure. So you can do stuff like that. You can create 3D models on that as well. So what you do is you take a slice with your focused ion beam, then you take an image and you take another slice with the focused ion beam and then another image, and you do this hundreds or thousands of times, and you can create this movie as you see here, or you can take all those images and create a 3D model. And so we've done work like that. This happens to be basically a semiconductor electrical circuit, and we're looking at tungsten posts and where they're positioned. So you can also use this to create a sample for the TEM. Remember, TEM has to be a sample for the TEM has to be very, very thin. And so something like a semiconductor, that can be problematic. And so, for example, we wanted to look at a semiconductor, and there's a little defect. If you can see that little kind of hole right there, we wanted to look at that defect in the TEM. So you can use the fib to create a trench on each side of that defect. And then you can actually cut down here and here and totally remove it and grab it with what's it's a micro manipulator. You can think of it as maybe kind of a tiny little tweezers. And so you can take your section, put it onto a TEM holder, and then put it into the TEM. Here's low mag images of the TEM. Here's the high mag. And so there's the cross-sectional image of this original defect that we wanted to look at. And so that's a neat application as well. All right, so just finishing up. The SEMs we have available, we've got an 840, which is a tungsten based. This is, again, if anybody is interested in taking the course, you'd start off on this one before moving on to our higher resolution instruments. But this also has EDS, and again, is a very capable scope, and usually for a lot of people in the class, that's all they need is this microscope. We have this little desktop microscope. The whole microscope sits on a desk like this, which means it's vaguely portable. So I'll take this microscope out to schools for educational events and show students and SEM. It's kind of neat. It's got some advantages. The main disadvantage is, excuse me, you lack resolution compared to my other SEMs. But 30 nanometers is still pretty good. And you don't have to do a lot of the sample prep that you have to do with my other conventional SEMs. This is similar to what's called a variable pressure or environmental SEM, which allows you to look at materials uncoated, for example. I don't have to coat them. I can look at vaguely wet samples. There's still a vacuum, but you can put a wet sample in and it will be continuously being dehydrated. But if you wanted to image very quickly, you can get information out like that. So it's definitely serves its little niche in the lab. And then finally, this is our high-res instrument. This is the one that's a dual beam, has both a focused ion beam as well as a SEM. It's got the EBSD system, EDS. So this is for the chemical analysis, elemental identification. This is for crystals. It's got a micro manipulator for doing things like creating TEM samples, things of that nature. It's kind of our workhorse microscope and our best microscope we have at the moment. So kind of summing up, TEM and SEM, they both use the same sort of hardware. So, you know, they're both using electrons, so they both use the same sort of electron sources. They both use the same sort of lenses, but their image construction is quite different. TEM is just essentially copying or taking the transmitted light microscopy idea and running with that. And SEM is more a probe technique. One looks at the surface. So here's two images of blood cells, for example, and one is looking at the surface. So you kind of get this 3D idea, whereas one is looking through the material. Both are useful depending on what you're trying to gain. And I'll just quit by saying we have these courses available that if anyone's interested, we offer an advanced light microscopy taught by Dr. Ting Gu, and TEM and SEM, which is typically taught by me. So in addition to the lecture component, learning about the instruments, you also get trained on the instrument. So it's kind of a neat way. If you have a semester where you know you're going to need a lot of SEM or TEM, it's a good idea to take the course at that point, because then you get a whole semester's worth to work on your research as you're learning the instrument. So with that, I think I'm probably a bit over, so I'll go ahead and stop at this point.
Thank you.
Okay. Yep.
Thanks, Dr. Preston. It was really an enlightening seminar.